Assalamu alaikum everyone and welcome to another ATP video. In the last two videos we talked about Pseudomonas, E. coli, and Shigella. Today we're going to discuss the fourth gram-negative bacteria on our list, and that is Salmonella. Salmonella is a gram-negative rod. She is motile, non-lactose fermenter, oxidase negative, and can produce hydrogen sulfide. Salmonella species grows in colorless colonies on MacConkey's agar, since they can't ferment lactose. And since Salmonella produces hydrogen sulfide, Salmonella species will grow in black colonies on Hectowen's agar and TSI agar. Remember that I told before that Shigella does not produce hydrogen sulfide, and so Shigella species grow in green colonies on Hectowen's agar. Now let's talk about the virulence factors. The first one is endotoxin, and by now you should figure out why she has an endotoxin. And the second one is VI capsule, which allows the Salmonella to survive the complement-mediated damage. Okay, let's move on to the clinical part now. First we have salmonellosis. Now there are two types of presentation depending on the species of the infecting Salmonella, which are typhoidal or non-typhoidal. Typhoidal salmonellosis, also known as typhoid fever or enteric fever, is caused by Salmonella typhi, and it's characterized by fever, constipation followed by diarrhea, and occasionally rose spots rash. When S. typhi is ingested in large amounts, she begins the infection at the ileocecal region and results first in constipation. Salmonella typhi has a special mechanism that allows her to get into the intestinal cell, but we will not go into the details of that today. In the first week of infection, Salmonella typhi invades the bloodstream, which will cause systemic symptoms, mainly fever. Remember, that's why it's called typhoid fever, and may also result in rose spots rash. During this period, the blood culture usually will be positive. It is important to know how S. typhi spreads through the bloodstream. This happens by invading macrophages, and once inside the macrophage, S. typhi escapes the killing mechanism and replicates. After that, S. typhi leaves the macrophages and infects the biliary tract, or the liver and gallbladder, in other words. And from there, S. typhi enters the intestinal tract again through the bile. And all of this happens during the third week of infection. And at this time, S. typhi causes diarrhea, and thus stool culture is usually positive at this point. Now, there's also a milder form of typhoid fever, which is called paratyphoid fever, and this is caused by another species called S. paratyphi. And that's it for typhoidal salmonellosis. Now let's move on to the non-typhoidal type. There are two common non-typhoidal species, S. enteriditis and S. typhimurium. It's pretty hard to pronounce these names. These species invade the ileocecal region, and after the invasion, they will cause inflammatory response by the immune system, and that will be associated with an increase in cyclic AMP. This will cause watery diarrhea first, but also these species can cause shallow ulcers, which will eventually lead to bloody diarrhea. Last thing we want you to know about in the clinical relevance is that Salmonella is considered the most common cause of osteomyelitis in sickle disease patients, not the trait. To complete our discussion about Salmonella, we need to talk about the transmission and vaccination. For Salmonella typhi, it's only found in humans, so it's transmitted by fecal-oral route. For the non-typhoidal ones, they are found in animals as well as in humans, and they are mainly transmitted through chicken products, but they can be also transmitted by reptile pets like snakes and turtles. Lastly, we have live attenuated vaccine for Salmonella typhi, but on the other hand, we don't have a vaccine for the non-typhoidal type yet. So maybe in the future you'll find the vaccine for it. Finally, it's time to put all of this together